Leslie, and Hannah.
Thank you.
Hi, everybody.
Thanks for coming today, guys. We're going to talk about the Drosophila melanogaster and how it's a common fruit fly. Did you know that the Drosophila melanogaster is a model organism and that it actually has a short lifespan, and that's why it's easy to maintain within labs? A lot of biologists use them to study the chromosomes and genomes and stuff. So they have a small number of chromosomes, and they can be used in research for human genetic disorders.
That's really interesting. I actually read something recently about T.H. Morgan and the first experiments with the common fruit fly, and he actually did a cross between two red-eyed flies, and he was studying them for possible mutations, and a mutation did come up, and it turned out to be a white-eyed fly, and it was only a male. There were no white-eyed females from this cross. So he decided to use that fly with the mutation and do another cross with a white-eyed male and a red-eyed female, and this resulted in both white-eyed males and females. This led him to conclude that it was probably a mutation in a single egg of a female that led to the production of a white-eyed male and female. And speaking of mutations, there are actually multiple mutations in the common fruit fly for their eye color, but primarily what we're going to focus on is the white and the sepia eye mutation. So the wild type eye color is actually red, and then two of the mutations that are very common are white and sepia eyes, and our focus today is sepia eyes. The types of inheritance for these mutations is autosomal and sex-linked, and specifically sepia eyes are autosomal and white eyes are sex-linked. And the type of mutation for a sepia eye color is a frameshift mutation that creates a stop codon, and the stop codon is actually TGA, but that's very specific. We won't get into too many specifics.
You know what this reminds me of?
What?
Have you seen that video of Drosophila?
No, I don't think I've ever
Oh
seen
my God,
that.
Hannah, we gotta watch right now.
Welcome to today's show, everybody. With us here today, we have B. Welcome, B.
Hi.
So, B, what brings you here today?
Well, Drosophila, me and my boyfriend have been together for 2 weeks now, and we even have a son that's 1 week old. But he doesn't think it's his and won't claim him because he has sepia eyes.
Well, you've come to the right place. Our professionals have taken a DNA test, and we have the results for you today. How's that sound?
That sounds great, Drosophila.
Well, let's bring out your boyfriend. Buzz, come on out. How are you doing today, Buzz?
I'm fine, Drosophila. How about yourself?
I'm doing quite well, but I know someone who isn't. B here, and I assume you know why.
Yeah, I know why. Because that baby has sepia eyes, and I don't think it's mine.
He is yours. Why don't you think he's yours?
Obviously, I have red eyes, but that baby has sepia eyes. Drosophila, explain this to me.
The results are in. In the case of this sepia-eyed baby, Buzz, you are the father.
What?
I told you.
What? How is this possible? Explain this to me.
Well, Buzz, for sexually reproducing species, we typically have 2 copies for each gene. These might be 2 dominant, 2 recessive, or 1 of each. If you have one of each for eye color, you'll only express the dominant trait. Sepia is an autosomal recessive trait that only shows up if both parents carry that recessive gene. While you and your beloved bee both express the red eye phenotype, you are heterozygous for the eye color genotype, having one dominant and one recessive allele. Red is the dominant allele for this genotype and sepia is the recessive. You both passed on one recessive trait to your offspring, giving him sepia color eyes. So don't worry, Buzz. He's all yours.
The materials that are needed in order to conduct this experiment is the F1 generation, which was the offspring of the mutant female showing sepia eyes and the wild-type male showing red eyes. As you can see in a picture on your right, there are brushes and magnifying glasses which were used in order to separate and help identify the sex and phenotype of each fruit fly. Fly nap, which was used to safely put the flies to sleep in order for us to count them. Plastic tubes and sponges in order to create a controlled environment for the fruit flies to breed in. The fruit fly medium, which of course is the food needed for larvae and pupa to grow. And netting, which provided a place for the fruit flies to rest and lay their eggs on. And of course, all of these materials were provided by Carolina Biological Supply Company. I do want to note that the methods took longer than they should have because it should have ranged to 3 weeks total, but in our experiment it was 4 to 5 weeks total because the F1 generation did not emerge the first week. But this is an overall general view of how the procedure should have gone, and it did. So the first week, we counted the F1 generation, which of course is the mutant female and wild-type male's offspring. We looked at the sex and phenotype of each fruit fly, then created a culture vial from the materials listed before in order to set up the F1-F1 cross. We then counted and put approximately 6 to 10 males and females into each vial. In week 2, we removed the F1 generation because they have already bred and created the F2 generation, which was in its pupa and larva stage. We did not need the F1 generation, and we essentially disposed of them. And we also did not want the F1 generation crossbreeding with the F2 generation. By week 3, the F2 generation emerged, and we were able to count the sex and phenotype of each fruit fly. And of course, the exact procedure is located in Carolina Drosophila Lab Manual. The results of our experiment were conclusive with what we expected. As we can look and see from the F1 generation, there is no sepia eyes present in either male or female. There are predominantly red eyes. Reason being is that the F1 generation is a carrier of the sepia eye color. So then when we bred an F1 with another F1, we then get the F2 generation, which of course you see there is still red eyes present. We predominantly see a lot more sepia in both male and female due to the fact that this trait is now being shown.
For our results section, we did a chi-square test. Chi-square is used to predict how close your data is to your hypothesis. We hypothesized that there would be a 3-to-1 ratio of red-eyed versus sepia-eyed flies. We expected for there to be 154 red-eyed flies and 51 sepia-eyed flies. This was calculated by taking the total number of flies, which was 205, and multiplying that by 3/4 or 1/4. We observed that there were 148 red-eyed flies and 57 sepia-eyed flies. We then calculated the chi-square by taking the observed minus the expected, squaring that, and then dividing that by the expected. The red-eyed chi-square resulted in a 0.234, and the sepia-eyed chi-square resulted in 0.706. Therefore, we added the two to get a chi-square total of 0.94. When looking at a chi-square distribution like the one shown here, we look at the p-values. A p-value greater than or equal to 0.05 resembles that the results were not significant. However, If the p-value less than or equal to 0.05, this resembles that the results were significant. Since we have a chi-square of 0.94, we fall between the 0.5 and the 0.25 column, because we only have 1 degree of freedom found by 2 minus 1, which equals 1. Since we fall between the 0.5 column and the 0.25 column, we have a p-value less than 0.05, which means that the results that we obtained were significant.
We hypothesized that there would be a 3 red-eyed to 1 sepia-eyed ratio. Our chi-square value after our experiment was found to be 0.94, and this resulted in a p-value between 0.5 and 0.25. This meant that our observations were significant and supported what was expected. This directly relates to Mendel's experiments with pea plants, in that our F2 generation displayed a 3 to 1 ratio. This is shown by the following cross, where the two individuals being crossed would be individuals from the F1 generation. As you can see, they're both heterozygous, displaying the dominant phenotype, in this case being red eyes, but they both carry the recessive phenotype, being sepia eyes. When you cross these individuals, you end up getting 3 that display the dominant or red-eyed phenotype, and 1 that displays the recessive or sepia-eyed phenotype. This directly supports our hypothesis of a 3 to 1 ratio.
Sources of error could be miscounting the amount of flies, incorrectly determining whether the fly was male or female, and if the fly had lighter sepia eyes, and the F1 and F2 cross. Some future research could be making an F3 generation and observing this ratio, or reversing the genotypes of the parent generation, so the mother would be wild type and the dad would be mutant.